Okay, Assalamu alaikum. My name is Nimra Shakeel, and I have joined Valeem as a biology and chemistry instructor. So today I'll be giving my lecture on the topic of cell, which is the chapter number 1 for the people who are doing intermediate or matriculation, and it is chapter number 3 in the books of OA levels GSCE. I'll be sharing the screen here. And I'll be starting with my slides. Okay. Without wasting any further time, I think it's visible to everybody. So it's for biology O levels in matriculation by me, and it's chapter number 3 according to the books of the O levels. It's cell structure and function. Today we'll be catering the introduction. So here's my brief introduction. My name is Nimra Shakeel, and I'm a 22-year-old girl. Currently, I'm enrolled in Fatma Jinnah Medical University, Lahore in MBBS program. I'll be completing my batch in 2021. Currently, I'm a 4th-year medical student. I'll be having my exams, and then I'll be in final year, and by 2021, I'll be graduate. So I did for like an insight into my educational career. I did my matriculation from a local school back in 2014, a school near my house, and I secured 1,035 marks out of 1,100 in my matriculation, and then I got admission in Lahore College for Women University. It's also situated in Lahore, and then I opted for FSC pre-medical, and I did very good in my FS examination. That was really good. It was 1,011 out of 1,100. Then in 2016, I took my SATs, and I got enrolled in LUMS in BS biology program. And at the very same time, I took my MCAT as well, and I secured 90% marks in it. And then afterwards, I decided to pursue my career as a doctor, and I left LUMS and I joined FJMU in the same year. I joined LUMS in August, and I guess in January, I joined FJMU to become a doctor and serve my country, which has always been a dream. I have been teaching people since 2015, when even I was a student. I have started this work since then, and there are a lot of students. Even some of them were my age group fellows, and some of were my peers. Some of them were in the same class as I was. So now it's been 5 years, and all of my students, alhamdulillah, they're currently enrolled in best of the best institutions of Pakistan. And I really hope that aap mujhse kuch seekhenge. I hope I'll prove to be a great help to my students at Valeem as well, because I've joined this just now. So before wasting any further time, let's continue with the introduction to the cell basics. So in today's lecture, I'll be catering the introduction to the cell and the cell history, cell theory and its timelines. Then we'll be talking about a little bit about cell biology, its microscopy. Then we would know how to use microscopes. Then there will be types of microscopes. It is very important because even if at FSC level, if you don't know how to use a microscope, even at the O levels level, I don't think somebody is going to teach you that in your graduation, when you will be, if you'll be pursuing medicine as your career. So isko abhi seekh lenge, so it would prove to be a great help. Then we'll talk about the sizes of cells, the classification of cells, the prokaryotes and eukaryotes. This won't leave you until you complete your graduation, post-graduation. Prokaryotes, eukaryotes, and the basic structure of cell, it should be known to everybody. So let's continue with the cell structure and its fundamental, cell structure. Cell is a fundamental unit of life. It is a self-replicating structure. What do we mean by self-replicating? Self-replicating means the thing which grows or which divides on its own, right? For example, if example आप इस तरह ले लें कि there is a human being and then a human being produces another human being. After a period of some time, we produce another human being. So इस तरह से generations to generations जो है चीजें चलती हैं. अगर आप एक bacterial cell लेते हैं, single cell की example देते हैं यहाँ पे हम. If you take a bacterial cell which is a single cell, if you keep it in a proper medium and a proper culture, after 20 minutes you will see the cell has divided into two, and then after 20 minutes the two cells have been divided into the four. So it is a self-replicating structure. It does not need any stimulus. It divides on its own. The first cell, it was discovered by a scientist whose name was Robert Hooke in 1665, from a piece of cork. When he observed it under his microscope, he took a cork and he made a slide out of it, and he observed it under his light microscope, and then he examined. If we see that diagram clearly, I don't know if I can zoom it, or if you can see it, you would see these little windows in it. He called it cellula, which means the compartment. To him, it looked like a compartment. Honeycomb-like structures hain, which if you can examine the diagram, see the pore-like regular structures that look like empty boxes surrounded by a wall. So he called it cellula. After like almost 200 years later, another general cell theory emerged from the work of two German scientists. They are very important. We call them the father of biology, Robert Hooke, Schleiden, and Schwann. Schleiden was a botanist, and he suggested that all plants are made up of cells, and Schwann was a zoologist. Schleiden put his work out in 1838, while Schwann, he did his work in 1839, and he suggested that it's the same phenomenon that goes for the animals as well. So let's come to the cell theory. The cell theory, it states that the basic unit of structure and function of all living organisms is the cell. It was proposed by Schleiden and Theodor Schwann. Matthias Schleiden and Theodor Schwann, these were the full names, in 1839, along with the other scientists. So here is a diagram that cell theory is a triangle, and it was produced by the work of three scientists, that's Schleiden, Schwann, and Virchow. So all living things are made up of cells. Obviously, every human being is made up of cells. Every plant is made up of cells. Even if we take bacteria, they're single cell structures, but still they have cells. Cells are the smallest working unit of all living things. Smallest working unit means, if we see a cell, you would see that every cell has its own powerhouse, which is providing it with all the energy. We have a brain of the cell, which is a nucleus, which is controlling all the functions, and which is controlling the replication, the functions of each and every organelle of the cell. Then we have the endoplasmic reticulums that are preparing the food. Then we have the lysosomes in a cell. So obviously वो waste management के लिए है. तो cell एक proper एक छोटा सा घर है जिसके अंदर different उसके members हैं, and every member is doing its work. Cell is like a factory. So हर member अपना काम कर रहा है to make the cell survive, right? Then all cells, they come from pre-existing cells through cell division. Cell division भी एक बहुत बड़ी branch है जिसके बारे में हम आगे पढ़ेंगे. So ये इस चीज को मैं पहले explain कर चुकी हूँ. I won't waste any more the time on it. Let's get to this cell theory timeline. In 1674, there was scientist Anton van Leeuwenhoek, and he observed a living cell. Before him, Robert Hooke, उसने अपना काम दे दिया था. He discovered cell. Whatever Robert Hooke discovered, it was a cork. So cork जो होता है वो तो dead material होता है. So he discovered dead cells. So he couldn't see, and he didn't have such specialized microscopes, so he couldn't see all the structures inside the cell. Then in 1674, about 10 years later, Anton van Leeuwenhoek observed living cells. And then after that, in 1833, Robert Brown, this was this person who discovered the nucleus. He saw a darkly stained structure inside the cell in the very center, and he called it nucleola, and then further it was called nucleus. So there is this cell theory timeline. Different scientists work on it. First of all, Felix Desjardins, he discovered the fluid content of the cell, which we today we know it as cytoplasm. Then in 1938, Schleiden, and then 39 May we had Purkinje and Schwann. Schleiden discovered that all plants are made up of cells. Schwann discovered all animals. In between, Purkinje, he named the fluid content of the cell as protoplasm. Today we call it cytoplasm. Then we have in 1845 Hendrik Braun. He proposed cell is the basic unit of life. And then in 1855, Rodolphe Virchow, he basically told us that all cells, they arise from pre-existing cells. So you see, there is complete timeline, एक पूरी chain है from scientist to scientist, और कितना ज़्यादा अरसे का वक्त लगा है, and then the cell theory was proposed. Now we have, how can we see cells? So we can see cells through a device that we call microscope, where micro means small and scope means to see. So the device which is used to see very small objects, very, very small, micro means very small objects that couldn't be seen with the naked eye. We use specialized instruments for them, we call microscope. It is a feature of magnifying the objects. Like magnify means to make an object look bigger. For example, make an image with a compound like microscope. It takes a power of the objective lens, which is 4 times. 3 lenses होते हैं in a microscope. If I could show you a microscope, अगर मेरे पास अभी हो तो मैं आपको दिखा देती हूँ, वरना इस पिक्चर में बड़ी क्लियरली है मेंशन है. मैं स्लाइड्स बाद में वलीम के प्लेटफॉर्म पे शेयर भी कर दूंगी. You can see it there. We have 3 lenses, 4x, 10x, and 40x. X means, right? X means times. So 4 times आपको image को magnify करके जो lens दिखाएगा, it's a 4x lens. Then we have 10x and 40 times the lens which can magnify the image by 40 times. It's a 40x lens, and we then multiply it by the power of the eyepiece. Eyepiece, actually we have it like this. It's like a tube, and we are seeing it like this. 2 lenses लगे होते हैं. One is near your eye, which is called the eyepiece, and one is near the object, which is on the stage, microscope के आगे. That lens is called the objective lens. The lens near the object you are viewing is the objective lens, and the lens near your eye is called the eyepiece. And then we multiply both of them, जितनी eyepiece की power है and जितनी objective lens की, and then we get the whole magnifying part, कितना उसने उसको magnify किया. Then there is this high resolving part. What is resolving part? It is the ability to see 2 things as discrete images. If we put 2 dots extremely close to each other on a notebook, on a page, on a notebook, and you can see the point where those 2 dots are separated. It's the resolving part of your eye. If I put 2 dots like this, and you can see those dots as 2 separated dots. Look, if you see it, यहाँ आपको थोड़ा separated नजर आएगा. तो this is the resolution. ये बहुत बड़ी example मैंने आपको दी. Dots अगर आप लगा के देखें तो आपको उससे resolving part concept जो है बहुत अच्छा समझ आएगा. With normal vision, there is a resolving power for 100 micrometers, which means 2 dots, 2 little objects who are 100 micrometers apart from each other, they can be seen as separate objects by the naked eye of a human being. Then what is cell biology and what is microscopy? So the study of cell, हम cell cytology कहते हैं, cytology, but at your level you can call it cell biology. The study of cell had given rise to an important branch of biology. It is known as cell biology or cytology. Cells now can be studied by using various types of microscopes. There are 2 fundamentally different types of microscopes now in use: the light microscope, the electron microscope. The light microscope means in which we are using light, and the electron microscope in which we are using a beam of electron. Both use a form of radiation in order to create an image. And the light microscope uses light as a source, and the electron microscope, it uses a beam of electrons. This is a complete picture of microscope. If you can see, there is this eyepiece, then there is this head, this is the objective lens, 3 lenses hai. You can see this is the stage clips. It is where we put the slides. This is the screw from where we adjust the control over the resolution and magnifying part. This is the aperture illuminator. यहाँ पे bulb होता है, वहाँ से हम light adjust करते हैं. And this is the electron microscope. ये बहुत बड़ी होती है. Or we put the specimen inside, then a beam of electron is passed through, and the image is formed, and it is projected, and the image is captured on the computer screens, and we can see that. Now we have a difference. This is very important according to your exam point of view. If you know the difference between electron and light microscope, for the people pursuing matriculation, this is an important question. Light microscope, it is commonly used nowadays. Visible light passes through the specimen. It is bent through the lens system. I have told you, यहाँ पे तो illuminator लगा होता है, यहाँ पे bulb होता है. In lens system, we adjust, mirror लगा होता है, so we adjust the light. It producing magnifying image. It could magnify by 1,000 times. It uses blue light at 400 nanometer. Lowest resolution is 200 nanometer, right? And then there's electron microscope. We use electron because it has a shorter wavelength. X-ray is not used. Electrons are used. Why X-ray is not used? Because it is very hard to be controlled. Electron is used. How electrons are controlled? So they are controlled by using magnets. There are two magnets side by side in an electron microscope. Electron can only function in vacuum. Oxygen molecules may cause the electron to be knocked around. So which atom is used to knock around the electrons? It's the oxygen. And then it is falsely colored by the computer, so we can see the desired structures. There are further two types of electron microscope: scanning electron microscope and transmission. In scanning, the electron beam goes, it touches the object and it comes back. So it gives us a superficial image, the outside image, a 3D image. Resolution is smaller than transmission, electron microscope. What is transmission? Electron passes through the surface, so हमें inner structures जो है अंदर से देखने में ज़्यादा help मिलेगी. For example, this is my hand. An electron beam is coming. It came and it just touched it and it came back. So the structure which is projected on the computer screen would be of this thing, not what is inside my hand or not what is there. But if the electron beam passes through it, so the image formed will be of all the structures, this side, what lies inside, what is here. So complete image ke liye. This is not very important, but this is for your comprehension. That a red blood cell, it sizes between 7 micrometer to 10 micrometers. Then we have egg cell, 100 micrometer. Egg is the biggest cell. Virus, it's 20,400 nanometers. Prokaryotes, this is the size. Nucleus is 6 to 7 micrometer. Ribosome is 24 nanometer. So you can see kitna chota size hai in micro and nanos. Level of cellular organization. This is very important. How the cell is formed. First of all, there's the atom, a very small structure. Atoms combine to form molecules. Molecules combine to form compounds. Then from compounds, different structures form. But at the level of biology, at the level of cell, then it forms the organelle. Different organelles, they combine to form a cell, a complete structure. Then different cells, they combine to form tissues. Tissues form organs. Organ forms organ systems. And then there's an organism. For example, there's an atom. It formed a molecule, it formed an organelle. Different organelles are there. There's mitochondria, ribosome, endoplasmic reticulum, nucleus, then different organelles are there. So many. I'm not mentioning that. I'm only mentioning that which are important at your level. And then it forms a cell, right? The cell then forms an organ, for example, which is stomach. Stomach cells bana diye usne. So then stomach cells and cells related to that, some of them, they form intestine, pancreas, liver. Every cell has a different structure and different function. So they form a complete gastrointestinal tract. Then there is some cells who will form the cells of kidney. So they will form kidney, bladder, and they will form your urinary system. So there is a complete organ system. And then a human being is formed, which has different structures. Hamare andar there is our excretory system, gastrointestinal system, urinary system, cardiovascular system, respiratory system. So different systems, they've combined together to form cells. If you see the diagram, the atoms, then there's molecules, cell, tissue, organ, body system, organism. Different organisms are combined to form a population. At a bigger level, population makes community. On the level of Earth, we make ecosystem. And on the level of planets, we make a biosphere. How do we classify cells? Very important question, very, very important from the paper point of view. Organism, it could be prokaryote, it could be eukaryote. In prokaryote, there is single cellular structure. इसके अंदर हम older organisms को लेके आते हैं, archaea and bacteria. Then we have eukaryote. Eukaryotes can be single cellular, they can be multicellular. Single cellular, we have kingdom Protista, then the protists. Then multicellular, we have plants, animal, and we have fungi. Then we have kingdoms of living things. In eukaryotes, we have animals, we have plants, we have fungi, we have protists, and in prokaryotes, we have bacteria. This is a repetition. It's just for you to see the diagrams. Then what is prokaryote and what is eukaryote? Definitions are really important. So any of the organism that lacks a distinct membrane-bound nucleus, it is known as prokaryote. Pro means before, karyote means nucleus. So U means true, karyote means nucleus. So one with the true nut or true nucleus is a eukaryote. So single celled or multicellular organism जिसके अंदर membrane bounded nucleus होगा, it is a eukaryote. Very important difference between prokaryotes and eukaryotes. A prokaryote is a simple organism, for example, a bacteria, no nucleus, no membrane bounded organelles. It has wall, it has the cell membrane, but it does not have any membrane bound organelle. Common cell structures are plasma membrane, cytoplasm, DNA dispersed होगा, nucleus के अंदर packed नहीं होगा, no packed nucleus, no packed cell DNA in a nucleus, no packed chromatin, it is dispersed in the cytoplasm. We have ribosomes. In eukaryotic cells, they contain eukaryotic cells, we call them eukaryotes. They are found in plants, animals, fungi, and protists, range from 10 to 1000 micrometers in diameter. DNA is contained within a membrane-bound nucleus. They have membrane-bounded organelles. All the organelles are membrane-bounded. They have all the structures that prokaryotes lack. See, you can see here, the cytoskeleton, ये membranes, endoplasmic reticulum. जबकि ऐसा कुछ भी यहाँ पे नहीं है. यहाँ पे सिर्फ एक organelle है, ribosome. The detailed structure है, 3D structure है. If you see it, we have the cell wall, capsule, cell wall, cytoplasm. Nucleoid, the nuclear material is dispersed. It is not packed like this. And cytoplasm is there, ribosomes are there. This ये जो आपको बिल्कुल salt and pepper सा structure नजर आ रहा है, this is the ribosomes. ठीक है, cell movement के लिए we have flagellas. Then we have in eukaryotic cells we have aria, rough endoplasmic reticulum. We have ribosomes, smooth endoplasmic reticulum. This is for the protein synthesis, DNA and protein synthesis. Plasma membrane, flagellum for the cell movement. We have centrioles. This is centrioles are for maintaining the cell structure. Mitochondrion, for it is a cell powerhouse. Microtubules to make cell, to maintain the shape of the cell. Then we have the nucleus, and nucleus के अंदर we have chromatin and then nuclear envelope, nucleolus. So I think we are done here. The next lecture I'll be discussing with you the plant cell structure, the animal structure, and we'll learn about the organelles individually. Till then, take care, and I hope you apprehended my lecture. And if you don't understand anything, just note it down. Send me your queries at my Valeem profile, or I'll share my contact with you. You can email me your queries. So before the next lecture, we'll discuss your queries, we'll discuss the answers, and then we'll proceed. So you can contact us at here, valeem.com, and these are the contact numbers. You can ask for me from that, and then you can hire me as a teacher. Till then, I'm signing off. Allah Hafiz.